My name is Sokwan, and I'm also an intern. And today my speaker is feature selection and dimension reduction for single-cell RNA sequencing based on multinomial. As some of you already know, my research area is not the single-cell RNA, and I first get into this subject while preparing for the journal club. So feel free to interrupt.
So let us read the abstract first.
Does anyone need more time to read the abstract? No. And here's the guest sentence. So let us begin the presentation. So single-cell RNA sequencing is very popular. For example, like we have one cell. Then we kill the cell and we get many kinds of mRNAs, like mRNA1, mRNA2 to the mRNA-J. And what we do is we apply something called reverse transcript to make DNA from the RNA, obviously. And then we apply PCR to maximize the number, and we attach molecule identifiers to count the number of the expression of each gene. And this is only for one cell.
So if we
Perform this experiment for
many cells,
then as a result, we will get these kind of data, like for each gene and for each cell, we could count how many of the genes are expressed. Based on this data, we perform analysis, these have 3 steps, like normalize, feature selection, and dimension reduction.
First, as a normalize.
So for each gene, we will have a distribution, for each cells, how many number do they have that gene? So
For these distributions, we perform normalize
in order to make mean equals 0 and standard deviation equals 1.
And secondly, as a feature selection.
Because at the original data, there are about 100,000 genes. So the dimension is too high.
Can we go back?
Okay.
Everyone understand what this means? Normalization? And other people, except for health. What is the example of normalization in your life? In high school, you learned something. Do you remember? To make the mean 0, what do you have to do? Subtract the mean, right? Then to make the standard deviation 1, what do you have to do? After subtract the mean from each sample, divide by standard deviation, right? We call it the G-score. Do you remember? And that's one example of normalization. And then if you look at the original data, as a count of the gene, so it cannot have a negative value, right? Correct? So, but anyway, to get the mean 0 and standard deviation 1, we have to change it to the plus and minus thing. And then what it says is typical data, if we do the normalization, it has that kind of some bimodal distribution
Yeah, right.
that happen if we do the typical normalization. Okay? Yeah?
Apply any log transform here.
Yeah,
I will explain later.
yes, okay.
Yeah, not here. Okay, so that's about the normalization. And for the feature selection, like at the original data, like it contains like almost like 100,000 pairs of genes. So like the dimension is too high. So like in order to like pick about 2,000 genes, like we pick some of highly variable genes, meaning that like for some cells, those genes are expressed, but like for other cells, like genes are not expressed. For those genes, or like highly expressed genes, meaning like for every cell, those genes are expressed. We pick genes with high variable and high expressed, like about 2,000 dimensions.
So highly variable genes means across the cell. So yeah. Okay, let's explain this. So let's explain the basic structure of the data. So row is cell. If there are 1,000 cells, there are 1,000 cells. There are J types of gene. So cell 1 has like a 10 gene 1, 20 gene 2, 30 gene 3. So every cell could be different. So cell 1, cell 2, cell 3, cell 4, cell, in my body, gene 1 will be 10, gene 2 will be 20, something like that. And then highly variable gene means if we select the gene 1 will be different across the cell. That we call, if the difference is very, very different, every gene 1 is 10, 10, 10, 10, 10, we call lowly variable gene. So 10, 20, 30, 40, 50, 60, then highly variable genes. Why the highly variable genes are more important? Because anyway, our goal is we want to see which cells are different from each other. Okay? So then we need a gene which can distinguish the cell. That's why we're focusing on the highly variable gene. And then why we consider highly expressed genes then? Let's say among the what is this J notate here?
Maybe dimension.
Yeah,
Dimension of gene or cell?
for gene.
Or gene.
So maybe it says we only select 2,000 genes for satisfying this condition.
Yeah.
Among this, I
Yeah.
see. So highly expressed gene means the level of the genes are enough.
Yeah.
We don't want it too low, but it's a or condition, okay? So then among the 100,000 column, we may select the 2,000 column. Then we reduce the dimension of the data, right? Okay.
Yeah, this is the feature selection. And the third one is like the dimension reduction. Like the most familiar technique is called PCA. Like for the original data, like we pick some dimension like which expresses the difference between data, but I will explain about this later in detail. So RNA sequencing analysis includes normalized, feature selection, and dimension reduction. But the problem about the normalized is, like when we have the original data, how do we normalize this? First, like for each cell, we normalize the total sum of gene expressions. Like generally, by 10 to the 6, but it's not a constant. Why we are doing this, like,
What is a normalized total sum?
yes, right.
Can you explain what is a normalized total sum of the gene expression? Can you explain?
For example, like, suppose we have at cell 1, we have one number of gene 1 and 3 number of gene 2 and say 2 number of gene 3. Then how do we normalize this? 1 over 1 plus 3 plus 2 times 10 to the 6. Same as and also for other genes.
So anyway, you mean that the total sum of the gene, you
Yeah,
want to
right.
make 10 to the 6,
Yes.
regardless of the cell. Everyone get the point? Okay.
And the reason why we are doing this is for each cells, there are some variability of how much genes are expressed. And by normalizing this, like we remove that kind of variability. And after normalize, like we put log after plusing 1. The reason why we are taking the log is to reduce the effect of the abnormally high values. The problem of this normalization is, for example, like if the original count from the data was 0, then when we normalize, then that value will be also 0, right? But if the original count was 1, then if the value varies from the value of ni, but it is about the scale of 10. So the gap between 0 and non-zero increases from 1 to 10. But when we think of the original count was 2, then this value also has the scale of about 10. Which means the gap between non-zero values are decreased.
So yeah, everyone think about the log function shape. How does it look like? Rapidly increasing and then saturating. And then we multiply some number by 10 to the 6, that means where we are, very right side.
So when we look at the original data, the probability distribution is like these kind of forms. But when we do the log normalize, we get these kind of results. And based on this data, like we will make mean equals 0 and standard deviation equals 1. Like this is before. And as you can see, the gap between 0 and non-zero and the amount of non-zero data becomes distorted, which means like data is binarized. What I mean binarized is from the non-zero data, like there would be some information, but like due to the log transformation, it loses the information. Everyone get the point? So when we apply the PCA based on this normalize, this y-axis represents the dimension 1, which is the most relative dimension. And as we can see, the dimension 1 is highly correlated to zero fraction, meaning that the fraction of 0 is the most meaningful among the data.
So what is zero inflation?
Zero fraction means like from the original data, how much fraction does the 0 have? when we apply the PCA,
Mhm.
the PC1, the most relative dimension, is highly correlated to zero fractions.
I see. So that means the zero data in the raw data has very strong information.
Yes, at the current normalized. And when we perform PCA with two dimensions, we could see that dimension 1 and dimension 2 are also highly correlated with the fraction of 0. Here the red represents there are all zeros, and this blue represents like there are less zeros. And as you can see, reds are here and blues are here. So as a summary, the information from non-zero data is being distorted, and the information of zero fraction is very relevant in the current.
In this graph, what is the meaning of the point, each point? Cell or
Each point represents cells. Yeah. So this was problem due to the normalize. So what authors said was, can we use PCA without normalize based on the original data?
Can I ask a question?
Yeah, sure.
And why don't we just ignore the first PC and continue with other PCs?
As you can see, like this is the second PC.
Second PC is also highly correlated.
Yeah, exactly. Yeah, right.
I see.
So in order to use PCA without normalize, let us look at how PCA works. I guess many of you would already know how PCA works, but I'll just explain. So from the original data X, which is n by j, singular value decomposition decomposes this matrix into three matrices, which is U n by n, and sigma n by j, and W transpose, which is j by j. And the characteristic of this matrix is the columns of W and rows of U are orthogonal, which means they are orthogonal and the size equals 1. And the another characteristic is at the W, the columns of W are the eigenvectors of this matrix, which is equal to the covariance matrix if we assume the normalize. And let us assume like the eigenvectors at the column W are arranged from high to low value. Let us assume that. Then how PCA works is, first we choose the dimension L, which we want to reduce. And then from the matrix W, we pick the first L number of columns. That's it. Then we could get the reduced data, T, which has dimension L as we wanted. And this is calculated by the original data X times this data WL.
I thought that when we apply the PCA, we set the center of the data. We assume zero mean or
Here,
when
yes.
we calculate covariance matrix, we don't assume any zero mean. Stuff like that.
here, let us assume zero mean for the covariance matrix.
Or do we assume zero mean?
Yeah, here, right here.
So X, the original raw data X, does the mean of each vector is not zero, right?
Each column vector.
I think X should be the normalized,
Oh.
right?
Yeah.
X should be.
X is normalized data or raw data?
It says
Normalized.
in this study they don't do normalization.
Yeah, but like this is for original PCA. When we
Yeah, without normalization at
Yeah,
this
right.
point.
this data X is the X normalized.
Give me a second. So let's think very simply. I think now story is too complex. So original data. So everyone know what is singular value decomposition, right? Yeah, you should know, right? Minu? Yeah, you learned 2 months ago, right? Yeah, so singular value decomposition, so U, sigma, V, transpose, whatever. So what is the meaning of P selected here, J? So here is diagonal component, so there is some component and then, so anyway, 0. Okay, so if we select, let's say 3 columns here, okay, 3 column means we select the 3 diagonal component, we select the 3 row. Okay, then so let me think. X omega L. So think about it. What is the meaning of X times omega L? So omega L is we multiply
Vm.
V지 V의 L 곱한다는 거지. 여기에 what is the meaning of we multiply BL? BL is only this small portion. But what we know is this is orthogonal matrix. Orthogonal matrix means if we multiply self, it becomes 1. Others, it becomes 0. What is the result of this? How the matrix looks like? N by N matrix times small portion. How does it look like? I guess it will be something like got it? And then 1 over 1 multiplied diagonal will be only this part will be survived. And then only this part will be survived. So although people call PCA is X times omega L, but anyway, the result of X omega L is, you know that in the singular value because it's 100, 10, 1, 0.1, diagonal component is reducing, right? Among this, we select the important diagonal block, and then we only look at that part. I think that's the meaning of this. Make sense? So we use the 3 PC means among the diagonal component we choose up to third component. And then 4 PC means we approximate up to 4 component. That's the meaning of PCA. Got it?
Yeah, this was the idea of the PCA. And surprising thing is this T and this WL is the reduced data, which minimizes the reconstructed error. Meaning that from the original data, we reduce the dimension, and there should be some error. And what minimizes the error is this reduced data T. Does anyone need a proof?
What is the A?
Wait,
A? Which x minus x, we multiply transpose.
that's right.
Okay. It doesn't make sense. X is n by j, right?
That's right.
It's n by L.
Yes. Times L by T.
Then size becomes same. Okay. Gotcha.
If someone is wondering why we multiply WL transpose here, the reason is like WL times WL transpose equals the identity. So this matrix goes back from the projection to the original space. So anyway, PCA is a solution
Of a optimization problem.
This is the point. So if we write again, PCA is the solution of optimizing problem, which finds matrix WL, which minimizes this value, where Ti and Wj are the rows and columns from these matrix. And minimizing that error is equivalent to maximizing the log likelihood. Is there anyone who is not familiar with likelihood? No, okay.
What is the meaning of really maximizing the? What is that? Can you explain that normal distribution?
Okay.
So here, Xij follows the normal distribution. So the probability density function would be exponential minus sigma squared Xij minus would be this kind of form, right? Regular like the constants. And when we maximize the likelihood, we multiply the probability density functions. So when we take a log here, like this is this should have these terms. So maximizing this value means minimizing this term, which is equivalent to this term. I also note that like the product of probability density function equals the sum of log, which means this sum. So this is how PCA works. It is the optimization problem. So let us go back to our original data. Let us write Yij means the original data, the count of gene J from cell I, And let us write Ni equals the total count from cell I. And let me just write pi ij as unknown relative abundance at cell I, meaning that at that cell there are many genes, and these pi ij represents the abundance. How much are they in here? And when we write these kind of forms, we could know that the sampling of the data will follow a multinomial distribution, meaning that for all data, we pick yi1 number of gene 1, yi2 number of gene 2, and we pick yij number of gene j. And this is probability here. But in real biology, the Ni is very large, and pi ij is very small because there are too many genes. So in that assumption, this multinomial distribution could be approximated as a blossom.
Ni is very, very
Large.
large. And then, two of relative abundance.
Yes, right.
So means sum of pi ij is 1.
Exactly.
Let's try understand this multinomial distribution function. Everyone, does it make sense? Anyone who can provide an intuitive explanation about that distribution? Sokhan, can you make a very simple example? Let's say we have only 3 genes. And then can you explain what is the meaning of f of 1, 2, 3? Yeah, with the example.
Suppose like for one cell, there are 3 genes. And suppose like we count as 1, 3, and 2. And let us assume the unknown abundance is, for example, 0.3, 0.5, and 0.2. Meaning that for the cell, when we randomly pick gene, then about half of the probability we pick gene 2. And for like 0.3, we pick gene 1. At this assumption, the f of the number of gene 1 equals 1, and gene 2 equals 3, and 3 equals 2 is equivalent to amongst 6 genes we pick with this probability. This is the multinomial distribution. So intuitively, we pick 6 genes from cell 1, and the probability of picking 1 number of gene 1, 3 number of gene 2, and 2 number of gene 3 is equal to this equation.
So everyone is okay with this? Any concern?
셀의 언더라인 true가
그쵸.
각 진들의 개수가 있으면 각 진 전체 프로틴 개수 중에서 각 진이 몇 퍼센트의 그 프로틴을 뭐 차지하고 있는가에 대한 true 디스트리뷰션을 가정을 하고
그쵸. 그게 얘네들이죠.
그걸 가정했을 때 우리가 지금 옵저버한 데이터가 있죠.
예.
그 옵저버한 데이터가 나왔을 확률을 계산한 거예요.
그러니까 그래서
네.
is true일 때 yes. Yeah, you mean? Can you explain one more time? I think your explanation is very clear.
Okay. So maybe we can assume that
Why don't you go?
okay. So this is actually assuming
True relative proportion.
maybe this is maybe there's some true proportion of each gene
Yes.
among. So there are
30%, 50%, 20% is true.
maybe gene one
Is
is
30%.
this is 30%.
Yes.
And
Gene 5 is
Yeah.
2 is 50.
gene two.
Yeah. That's true.
Yeah, that's true proportion.
Okay.
And this is 20%. So this is 2%, but when we observe one cell, we observed one gene, observed 3 gene 1, 3 gene 2, and 2 gene 3. But generally they follow this distribution, but we observe this. Then what would be the probability that we observe this case?
Yes.
So this is what this F value means.
Yes. I think, yeah. Is it clear to everyone? Yes. So, anyway, Sokhan. So, and everyone here, so when you give a talk, and then when you tell about some equation, it's always good to show the one example. Then people will understand better always. Okay. Okay, then we finish up to the that equation. Then typical Ni is very large, and the 5ij is a point something, so Ni is pretty large number. The pi ij is small, then you follow the Poisson distribution.
Yep.
I see. So then can you write down the Poisson distribution with your example? So y then y1 will be, according to your example, y1 1, yeah,
Right.
I1
For cell 1 and for gene 1,
gene 1
each puzzle
Poisson
sum with
6 times 0.2.
yeah,
That is Poisson 1.8,
8.
right? So yeah, it makes sense, right? Think about it. In total, we have 6. The proportion here is 30%. Then what is the average of this gene count? 6 times 0.3, isn't it? 6 times 0.5. 6 times 0.2. Then each one has independent Poisson distribution. Actually, these three should be related each other usually. That's why we had a multinomial distribution, but I don't have any intuition, but if we say that Ni is large enough, and then pi ij is very small, then it looks like we can assume that every component has independent Poisson distribution. It's not trivial to me. Is there anyone this is trivial?
Are we considering they are independent, or this is just
But,
saying that marginal distribution?
oh, this is a marginal distribution, or I don't know. But if we
That-
assume that this Yij is Poisson, then this is just multiplication of this Poisson, isn't it? 그렇죠. 지금 독립으로 돼버리는 거 아닙니까?
네.
맞죠.
어,
그러니까 약간 저도 직관적이진 않은 게 ni가 크고 여기가 작으면 이 멀티노미알이 몰라. 난 수업 시간에 저거 배운 적은 없는데. 챗GPT에 물어봐?
근데 일단 화속을 맞추고 싶어 하는 생긴 게 쉐입이 생긴 게 생겼네.
Everyone, can you ask to ChatGPT? Multinomial distribution can be when the approximate is also. Large number of the trials, small probability then multinomial can be puzzle. Ooh. Yes. 와, 네. 왜 그런지 나도 모르겠는데. 일단 anyway, this is looks like well accepted. 석관, do you know why?
Not exactly.
반응음에 대해서 증명이 있는데
And yeah, let's try. Yes. So we will throw the dice. Okay. We will throw the dice n times. Okay. Then we have 1, 2, 3. So there are 6 categories, right? Got it? And then if we dice the 6 times, then maybe 1, 1, 1, 1, 1, 1, or 2, 1, 0, 1, 1, 1, 1. That kind of thing is possible, right? Why I'm talking about the 6 times means n is small now. If n is small, can we consider that all these are independent each other? I don't think so. If this is a 5, then it strongly affects the other parts, right? Because the total sum should be 6. Agree? But let's think about, we are not doing the 6. 10 to the 6. And then another condition here is the chance of each component is pretty low. The pi I is low. That means this number will be very, very small. Every number could be very, very small. And let's think about it. Among the 6 dices, not just 6, maybe there are many events, and then knowing him, among the 10 to the 6, knowing that the first, this first dice comes out 10 times, does it useful information to guess other thing? No, right? So that's why I think we can do the sum independency. So again, if we just the 6 time roll, knowing that here is 1 or 2 is very, very important information to make the guess about this, right? But if it's 10 to the 6, I'm not sure. And then let's say, but this is a very dominant thing, so that knowing this high probability, then maybe it can affect other things, but it should be low chance, it's a very small number, right? This knowing this low number chance may not affect other things. So that might be the rough reason why multinomial can be approximated as a Poisson distribution, but mathematically it should be easy, I think. Maybe you can just n times pi I is fixed, and then sending the limit of n to the infinity, then it will go to the, I think, Poisson distribution. But anyway, let's believe it. Every component has a Poisson distribution now. Okay.
Okay, so this was the point. So each entry is picked from Poisson distribution. Then why are we doing this? Because we want to use PCA. And let us go back to PCA. Like PCA was the optimization problem, which minimizes the error between original data and the reduced data. And this was equivalent to maximizing the log likelihood. And here, the original data follows a Poisson distribution. Therefore, what we should do is maximize the likelihood of the Poisson distribution. Everyone got the point?
Why do we call generalized PCA, not just PCA?
Because the method itself applies for every exponential family distributions, but here I am just focusing on Poisson.
So I see. So here new original PCA maximizing the normal distribution.
Yes, right.
But now here we want to maximize exponential Poisson distribution.
Yeah, right.
Okay. So previously we have a data, but we normalize it so that it looks like a normal distribution. And then we apply the PCA because it maximizes the likelihood of the normal distribution. But now we don't do the normalization, so I see we assume that original data follow the Poisson distribution so that our new PCA should maximize the Poisson distribution likelihood. Oh, clever. What do you think, everyone? Does it make sense? So typically in the step, to use the classical PCA, we have to make the original distribution to the normal. So that's why we perform the normalization thing. But that normalization caused the bias, so we skip that normalization, but we apply the PCA to the original data set. But that original data set followed Poisson rather than normal distribution, so our PCA will be changed. 똑똑한데? 이해됐나요? 괜찮습니까, everyone? 오, 야. 야, 근데 매우 쉽다.
In this case, the error function also minimizes.
Sorry.
Traditional PCA, the error minimizing error function equals maximize the normal distribution.
Yeah, right.
In this case, also it equivalent.
Yeah, right.
Okay.
I think there's a one limitation in this approach. Actually, so this assumes that every cells have the same, the pi I assumes pi I distribution, am I right?
Oh, no. Because pi_ij is different from each cell.
Ah, okay.
그러네. 똘똘한데? 근데 I think it's brilliant idea. Oh, it's very brilliant. Summary is let's call classical PCA. To use the classical PCA, we need a data should be basically we need it. So so real data. I don't know. It has some distribution. Then we cannot apply the PCA. So the first step we have to do is we did some pre-processing. Subtract the mean and then divide by blah blah. And then we can apply the classical PCA. The problem is this pre-processing can be make the distortion for the data. So we hate it. So that in here what we found is real data has Poisson distribution. Then rather than apply the classical PCA, we apply the generalized PCA, which is maximizing Poisson distribution rather than exponential normal distribution. Very smart. Not only for the single cell data, I think we can use it for anything. Okay. Everyone got the point?
And note that like from the original data. Oh yeah, sure.
Why the exponential function is in the Poisson distribution?
That's what I want to explain. So from the original data, we first normalize and we take the log. That's how we normalize it. And when we look at the mean of this Poisson distribution, it should be this one. And if we normalize, we divide it by ni. And we take a log, then this mean would be same.
Okay, we have to do normalization?
No, I'm just explaining the relationship between original data and normalization.
Original data, normalize and log distribution, then it become normal.
Yeah, right.
Okay. That's why they put the exponential.
Yes, right. But here, like note that like from the original PCA, like the reduced data t equals x times WL. So we only need to find WL. But here, we don't know the relationship.
Yes.
So we should optimize for both Ti and Wj here.
We cannot use the SVD then.
Yes.
So
So this is optimization problem.
I see. Classical PCA or singular value decomposition maximize normal distribution. see.
So because this is the optimizing problem, in order to maximize the likelihood of Poisson, we know that Poisson follows the probability mass function, like these kind of forms. So when we take a log, we will get this equation. Everyone clear? And here, Yij term is from the original data, so we could ignore this because we want to find such Ti and Wj which equals mu ij. So, in order to maximize the likelihood, the objective function should be the sum of log, which is equivalent to the multiplication of probabilities. Therefore, the objective function would be these kind of forms, which is exactly the same with this one. And I just put a new notation mu ij, which is exactly the same as this one.
So probability is that, and then if we apply the log, then that is it. So that data is gone, and then let's see. 그래서 UIJ가 뭐였죠?
그 여기 파라미터를 나타내기 위해서 그냥 따로 정의했습니다. 길게.
포화정 안에 있는 거?
예예.
오케이.
And this is the objective function, and we could optimize by using Lagrange multiplier or manifold optimization or like something else. So there are many ways.
So when we apply the generalized PCA,
So the first row was from the original one, and second one is from the generalized one. And at the original PCA, as we saw before, the fraction of 0 is correlated with the first PC. And the red one was here and blue one was here. So the data was correlated with zero fraction, which has a distortion. But when we use a generalized PCA, we can see that the fraction of 0 is not correlated with the first PC. It is these. So when we apply PCA,
1, 2,
we could check that red and blue are well mixed here, different from this one.
Very nice. Wow.
and when we apply the clustering by using k-means,
For two different data sets. This was from the generalized PCA, and this red one was from the original PCA. And I would say that generalized PCA is better because when you look at the second row, the purple and pink are not well mixed, but here, purple and pink are mixed. And also Dr. Kim said that generalized PCA is much better.
Actually, the performance of the GLM PC is quite amazing.
Mm.
But one cons is it's super slow.
exactly.
Yeah.
Yeah, because generalized PCA is the solution of the optimization problem, while the original PCA was just finding the eigenvalues and eigenvectors.
So when we look at the performance, the generalized PCA is 10 times much slower than PCA. This is the problem, but
Anyway, the result was good. So
I will summarize the take-home messages. So single cell RNA analyzes the cell, and PCA is nothing but the SVD, but it also minimizes the reconstruction error. But PCA has a problem at the normalization. Therefore, authors use generalized PCA, which prevents the loss of information. That's all for today. Any questions or comments?
In the generalized PCA, how we can select the how do we know which component will be more important? Since the original one we know that the components ordered and sorted by the singular value. But how do we know the importance of the generalized PCA?
Because generalized PCA maximizes the likelihood, which means this minimizes the error between original data and also the reduced data.
So, and actually, so if you obtain the T and W and the loading value, U in the loading value, right? The importance of the variance of the piece, each piece could be obtained from a T. So, yeah.
So maybe here, rather than singular values, you can measure how the likelihood increases as we increase the principal components. Then if that's saturated, then we can stop it.
이게 높은 저 알고 있는 U랑 V를 찾는 게
아, 그게 SVD가 아니라서 어렵다는 거.
이게 그러니까 단순히 그 매트릭스가 매트릭스 팩토라이제이션을 구하는 게 아니라 U랑 V를
옵티마이제이션 해야
구하려면
되니까.
미방을 계속 해야 되는데 저기 엘리먼트가 너무 많아서
I
굉장히 큰 데.
I see. So, it's beautiful, but the limit is clicks per PCA. We just need to do the SVD. Easy. But this one, we have to do the optimization. Wow. That's the problem. So SVD is really super easy. So that if we perform the SVD, then PCA comes on free. So we can do easily, but general PCA anyway. Oh yeah, here is computation speed. Yeah, and it's tenfold difference.
Yeah, right.
That's the problem of I think generalized PCA because it's really optimization.
So how many parameters we need to estimate? T and
handle the data dimension.
T and W for each. So,
TI. So I means that at least the number of
yeah, exactly.
number of cells.
Yeah, times number of genes.
Number of genes, not L.
Yeah, that's all.
뭐 말하시는 거야?
저희도 말이죠. 하는 이유.
안 해도 사실 되는 거야.
저희가 하는 이유는 싱글 밸리티 컴포지션을 쓰려고
싱글 밸리티 컴포지션.
PCA를 쓰려고
근데
정규화 제품.
근데 랜덤 매트릭스 띄우기는
가장 큰 문제가.
근데 랜덤 매트릭 띄오리를 쓰려면
예.
각각의 컴포넌트가
예.
어떤 조건에 평균이 0만 되면 됩니까? 아니면 노멀 디스트리뷰션 아니어도 되잖아요.
다 됩니다. 예, 다 돼요.
평균이 0이어야 되나?
아니 그렇게 다 되는데 문제는 뭐냐면 이거가 저희가 차원을 축소를 해서 축소된 차원을 오프인 하는 거잖아요. 저게 그래도 쓸 수 있는 기구가 10개 정도 되잖아요, PC.
아니 이걸 쓰자는 게 아니고
예.
그냥 우리가 그러니까 그냥 PC를 쓸 거면 알겠어요. 그걸 해야 되는 거.
예.
근데 우리는 시그널이 아닌 걸 고르려고 하는 거잖아요. 일단은.
예.
그냥 원래 로우 데이터에다가 랜덤 매트릭스 띄고 있으면 왜 안 되는 건가라는 생각이 들어서 여기서는 PC를 써야 되니까 뭐 그런 건 알겠는데
예. 원래다가
그냥 랜덤 매트릭 띄오리에서 그냥 아이겐밸류 벡터를 뭐 하면 그냥 되는 거 아니에요? 여전히 따라야 되는 거 아니에요?
그거는 맞죠. 그거는 맞지만 그냥 아예 아무것도 안 하고요. 그러면은 그 고질적인 문제가 베리언스가 처음에 나온 PC1의 베리언스 상당 부분이 하이리 매력을 주기 위해서 다 설명해요. 그러니까 결국 노멀리제이션을 해준다는 건 다 맞춰준다는 건데 그래서 되게 소수의 진들이 다 설명해버려요. 근데 뭐 원래 그냥 로그 데이터에다가 PCA를 적용해가지고
여기서는 이 제너럴라이즈 PCA는 사실 했을 때 그럼 그 이슈가 없는 거는 소수의 몇 개만
쟤는 근데 이제 뒤에 테일이 있으니까 큰 그건 한번 생각을 해보세요.
생각해 보시고 so everyone I don't know anyway this is bottleneck any idea can we convert this where is it optimization problem yeah we want to optimize this with the two parameter right actually it's not the two parameter it's many yeah it should be very difficult but if it is statistician should do something so yeah can we I don't know if someone can modify the SVD to get this, then it would be beautiful. So SVD is corresponding to the maximized normal distribution. Can we get the generalized SVD to get the generalized PCA? Then you can become immediately professor of the stat department. Seriously. Is it that difficult? Yeah.
네, 아이디어 자체가 다른 게 SVD가 애초에 노멀라이제이션 해야 되는 이유가 싱귤러 벡터가 이제 베리언스 매트릭스의 그 아이겐밸류라서 베리언스가 최대가 되는 데를 구하는 건데
맞아요.
애초에 이거는 노멀라이제이션을 안 하는 거라서 SVD를 제너럴라이즈 한다는 게 가능할지 모르겠습니다. 아이디어 자체가 달라서.
진짜 안 되나? 어려울까? 되면 참 좋겠는데.
근데 저기서 그냥 미분한 다음에 0으로 두고 익스플리시트하게 못 구하나? 진짜 원래 제너럴라이즈드 PCA에서
그게 라그랑지 멀티플라이어고
근데 그건 이제 그냥 라그랑지 멀티플라이어를 이제 알고리즘 상에서 적용한다는 거고
네.
저기서 그냥 익스플리시트하게 우리가 손으로 풀 수 없는 거예요. 그래서 포뮬러가 있으면 그냥 포뮬러에 넣고 계산하면 되잖아.
잘 모르겠어요.
WJ가 올소금을 한다는 가정도 필요한 거죠.
네. 그래서 라그랑지.
라그랑지.
화정도 더 노말이 될 수는 없지. 이거 없나? 일단 람다 크면 같긴 같은데 n이 크면 람다도 크면 근데 베리언스랑 민이 무조건 똑같으니까 벌써부터 리스트릭션인데 this general PCA idea is very lovely, but impractical. That's the problem. Too slow. Okay. Math department students. KAIST Post-Tech, Busan University, 서강. You can do it. 예, 대학 전쟁 할까? 이거 안 되나? 진짜 어렵겠지? 쉬우면 했겠지?
근데 모딩 값을 모딩 값만 구할 수 있어요. IM 밸류에 해당되는
그렇지.
그 베리언스 값만 구할 수도 랜덤 매트릭스를 쓸 수 있거든요.
그렇지.
다 그 벡터 다 구할 필요 없이 그 방법이 있을 수 있고.
진짜 아쉽네. It's beautiful but impractical. That's sad. By the way, any other question to Seok-Hwan? See, this is why every people always assume the normal distribution. Then life becomes easier. Yes. Accidentally singular value decomposition maximize the normal distribution likelihood or variance. Isn't it surprising that everyone say, I know the PCA, 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 but if distribution becomes just Poisson, we can't. Poisson is also extremely easy distribution, isn't it? Wow. It's really sad. think about a couple more minutes. So one option is we can modify the same SVD to get this work, but what else? What kind of any other idea to get the generalized PCA? Neural network, flow matching. Just think about whatever. Yeah, whatever. Think about all the weapons you have. Some distribution. Lots of. I always wonder why they multiply 10 to the 6. But now I feel why they do it. If we multiply 10 to the 6 to the lambda, then this becomes extremely large number. Maybe normal is okay. Quantum 10 to the 6 might be normal distribution, isn't it? Like this. Ah. 아, 이런 찝찝한 페이퍼라. 아름다운데 써먹을 수가 없다. 아, 이론은 진짜 좋은데. 야, 저 논어를 벗어나면 우리 못하는 거다. 아, 프러스트레이팅하네. 통계학자들 뭐 해서 이제까지. Anyway, think about it, and then if you come up with a better idea, discuss with the subclass. Okay, if you don't have any question, let's take the subclass. Okay, so Journal Club is finished earlier, so from now until 4 p.m. I have about 40 minutes for the meeting. Is there anyone